Tau protein case study by Nora Erickson and Joseph Caldwell.
Katie is a 50-year-old female that presented symptoms of forgetfulness and inability to name common items. Her coworkers recommended that she see a doctor. A doctor did extensive testing on her mental cognition before having a simple cerebrospinal fluid drawn and analyzed. It was found that Katie had an elevated amount of the protein tau in her spinal fluid, which coupled with her mental cognition test was enough to give a diagnosis of early onset Alzheimer's disease. Doctors cannot be certain of an Alzheimer's diagnosis until a post-mortem autopsy is done on the brain. Alzheimer's is a type of dementia that causes problems with memory, thinking, and behavior. Symptoms usually develop slowly and get worse over time, becoming severe enough to interfere with daily tasks. There is no cure for Alzheimer's disease, but medications and management strategies may slow down the progression of the disease. Most common cause of death in Alzheimer's patients are malnutrition and infection. Katie was diagnosed 5 years ago and is currently on the 6th stage of the disease. Doctors have estimated that she has about 1 year left to live. The exact cause of Alzheimer's disease is not known, but there are two biochemical factors thought to be related to the disease, called plaques and tangles. The buildup of plaques cause the formation of tangles, but we will only focus on the tangles, as this has to do with the tau protein.
Tau is abundant in neuron cells. It is not present in the dendrites of neurons, but is found in the distal portions of the axons. The function of tau protein is to help stabilize microtubules in the axons and provide flexibility. Tangles are found inside of the cell as opposed to plaques, which are found outside the neuron. Like other cells, neurons are held together by a cytoskeleton, which is in part made of microtubules. Microtubules are track-like structures that run the length of the cell in which nutrients and molecules are shipped along. Tau makes sure that these proteins stay in line and don't break apart. It is believed that the accumulation of plaques initiates the activation of kinase, which is an enzyme that transfers phosphate groups to the tau proteins. This hyperphosphorylation causes the tau to change shape and stop supporting the microtubules. The tau protein then clumps together with other tau proteins and leads to the second characteristic finding of Alzheimer's disease, which is neurofibrillary tangles. Neurons with tangles and non-functioning microtubules can't send and receive signals as well as healthy neurons and end up dying.